Welcome and good evening, everyone. Okay, so today we'll be talking about resting potential. All right. So candidates should be able to explain the formation of resting and action potentials. Now, first things first, let's recap what you have learned in Form 4 and Form 5. Okay. First thing is about impulse. Okay, so impulse is an electrical signal that involves in changes in potential difference across the surface membrane of neuron that sweeps along the neuron from one end to the other. Okay, this definition of impulse. Now, if you recall the direction of impulse travel, right, from dendrite to cell body, okay, and then along the axon, and finally to the dendrite of the next neuron. Okay, so previously we talked about the neural glia, especially the Schwann cell, right? How it formed the myelinated sheet around the axon. Okay, so now I'm going to show you this video. Credit to the owner of this video, right? About the Schwann cell and the action potential.
What you will see and hear in this presentation is the development of myelin in the peripheral nervous system and the propagation of the action potential along a myelinated axon. This multimedia presentation will be most helpful if you already have a good understanding of the Schwann cell and the electrochemical process of the neuron, called the action potential. The Schwann cell forms a protective covering around the axon. Schwann cells start to develop in the embryo and continue to increase the wrapping around the axon through childhood. This development increases the thickness of the wrappings, which peaks in adolescence. This is why teenagers have such quick responses. The Schwann cell contains the typical cell organelles and cell membrane structure. However, notice as the Schwann cell surrounds the axon that the nucleus and other organelles are squeezed to the outside wrapping of the cell. This outer wrapping of the Schwann cell is called the neurolemma. The inner lining is made up of layers upon layers of cell membrane. This inner wrapping is called the myelin sheath. You will recall that the cell membrane, called the fluid mosaic model, is made up of a bilayer of lipids integrated with proteins. The thicker the myelin, in other words, the more layers of cell membrane making up the myelin, the more advantageous it is to the axon. One advantage is the regeneration of severed axons. Another advantage is an increase in the speed of the propagation of the action potential along the axon. The rest of this presentation will concentrate on the increased speed of action potentials down the length of the myelinated axon. Here is the neuron, and you can see the repeated Schwann cell membrane forming the myelin. Note that there is a small space between the Schwann cells where the axon is not covered by the neuroglial cell. These spaces are called nodes of Rambier. From what you already know, action potentials occur at the axon hillock and continue to be repeated away from the cell body, much like dominoes falling one after another. An action potential starts on a polarized membrane, which is -70. A stimulus causes the sodium gates to open slightly, and sodium starts to trickle into the cell. If the cell reaches -60 or threshold, the sodium gates open wide and sodium floods in, bringing the inside of the axon to +30. At this point, the sodium gates close and potassium gates open. Potassium starts to pour out of the cell. This allows the neuron to become polarized again. Then the sodium-potassium pump starts to actively transport sodium out and potassium back into the neuron. First, we will look at the propagation of the action potential in the unmyelinated axon. Propagation is the repeating of action potentials down the axon. The action potential is repeated because as the sodium comes in, it diffuses to adjacent areas within the axon. As the sodium increases in this area, threshold is reached. Sodium gates open wide, sodium rushes in, causing depolarization and an action potential. As the sodium enters this area, it diffuses through the exoplasm, and another action potential is created. This continues down the length of the axon. Now look at the myelinated axon. The same process applies to the myelinated axon. An action potential develops, and as the sodium comes in, it diffuses through the cytoplasm of the axon. It continues to diffuse through the portion of the axon wrapped in myelin. The increased sodium concentration reaches the node of Ranvier, increases the axoplasm to -60, and depolarization occurs. The sodium gates open wide, sodium floods in, and we have an action potential. Again, the sodiums diffuse through the axoplasm, reaching the next node. An action potential develops. The process is continued down the myelinated axon, passing from node to node. Compare the unmyelinated axon with the myelinated axon. You can see that action potential reached the end of the myelinated axon more rapidly than the unmyelinated axon. The speed of the propagation is faster going from node to node than action potentials that develop adjacent to the previous action potential.
Okay, so the video already told you about the propagation of the action potential, okay, on the myelinated sheet and non-myelinated axon, myelinated axon and non-myelinated axon, okay, as well as how the potential difference across the membrane of a neuron that triggers the action potential. Okay, so now let's continue with resting potential. Okay, so resting potential is the potential difference across the cell surface membrane. You can see this is an axon. Okay, this is an axon, and this is the bilayer lipid of the plasma membrane of the axon. Okay, now the potential difference, okay, across the cell surface membrane of a resting neuron. This is the resting potential. Okay, the potential difference here or here, which will give you the value of -70 millivolts. Okay, now it is at a value of -70 millivolts, or you can say that the resting potential, right, of a neuron is -70 millivolt because the inside of the plasma neuron, right, or exoplasm is more negative than the outside or the extracellular fluid. Okay, why? Why is the exoplasm is more negative compared with the outside environment? Okay, this is because of there are three factors here. First is the higher number of organic acids and protein inside the exoplasm. Okay, and then the sodium potassium ion pump, okay, maintains the potential difference by actively pump 3 sodium ion to the extracellular fluid from the exoplasm and pump 2 potassium ion into the exoplasm. Okay, so you can see this sodium potassium ion pump here. Pumps 3, actively pump 3 potassium ion, sodium ion, okay, into the extracellular fluid and pump 2 potassium ion into the exoplasm. Okay, then there are more potassium ion channel, okay, than the sodium ion channel in the plasma membrane of a neuron. So meaning to say more potassium ion will diffuse out from the exoplasm compared with the number of sodium ion to diffuse into the exoplasm. Okay, another factor is sodium ion gated ion channel and potassium ion gated ion channels are closed, okay, during the phase, this phase in referring to the resting potential. Okay, so meaning to say when a neuron is resting. Okay, the potassium ion gated channel and the sodium ion gated channels are closed. Okay, sorry. Right, during this phase, in referring to a resting neuron. Right, so on the surface of a neuron or on the surface of an axon, okay, we have sodium potassium ion pump. Then we have um, sodium ion channel. You can see this is the sodium ion channel. Then we have the potassium ion channel. Okay, and we have sodium sodium gated ion channel and potassium ion gated channel. Okay, it is not shown here. Okay, so roughly this is how sodium ion gated ion channel look like. Okay, so 4 factors maintain a resting neuron, okay, in its resting potential, which is negative 17 millivolts. Now, how does an action potential form? Okay, okay. First things first, action potential of a neuron, also known as nerve impulse or spike. Okay, so when we talk about spike, we understood it means action potential or nerve impulse or impulse itself. Okay, action potential, okay, is the depolarization on the neuron plasma membrane. Okay, so meaning to say a resting neuron, okay, has a plasma membrane of being polarized. Okay, so being polarized into depolarized, it means impulse is being formed. Okay, so how does the neuron plasma membrane become depolarized? Right, okay, it becomes depolarized when sodium ion enters inwards, okay, into the exoplasm across the membrane, meaning to say across the exoplasma membrane, and change the potential difference of a resting neuron. Okay, so we have this graph here, membrane potential against time. So this is the part where we have a resting potential. Now, formation of action potential consists of 5 stages. Okay, resting phase, depolarization, rising phase of the action potential, falling phase of the action potential, and undershoot. So we go to the first one, right, which is resting state. Now, in resting state, what happens? Most sodium ion gated ion channels as well as the potassium ion gated ion channels closed. Okay, then some sodium ion channel and some potassium ion channels are open to maintain the resting potential. But earlier I did mention, right, that um, during resting potential, what happens? Sodium ion gated channel and potassium ion gated ion channels are closed. Okay, but these two channel, sodium ion channel and potassium ion channel, are open. Okay, to maintain the resting potential. So we have key here. Okay, this represents sodium ion and this represents potassium ion. And this is the sodium ion gated ion channel, okay, while this is the potassium ion gated ion channel. So during resting potential, these gated ion channels are closed. Only sodium ion channel and some potassium ion channel are open. Now, let's look here. Okay, the second stage, which is known as depolarization. What happens here? Okay, when the plasma membrane of a neuron is depolarized by a stimulus, okay, some sodium ion gated ion channel will open. Okay, causing inflow or influx of sodium ion into the exoplasm. Now, why sodium ion gated channel will open first? Because the sodium ion gated ion channel is more sensitive to the changes in the environment, or the sodium ion gated ion channel is more sensitive to stimulus. So when the stimulus, what happens is sodium ion gated ion channel will open and causing inflow or influx of sodium ion into the exoplasm. Okay, can you imagine? Can you picture that? Okay, some of these sodium ion gated ion channel will be open and allow influx of sodium ion into the exoplasm, while some remain closed. So you can see how this sodium ion bounces off. Say it doesn't enter or influx into the exoplasm, but some again will open and allows the influx of sodium ion channel into the exoplasm. This sodium ion channel remains open and sodium ion remains diffused into the exoplasm. So when there is an influx of sodium ion into the exoplasm, okay, what happens to the exoplasm? It becomes less negative. Okay, so initially it was negative 70 millivolt. So now less negative, it may reduce into negative 50 millivolt, negative 10 millivolt, for example. So the sodium ion inflow or the inflow of sodium ion will causes further depolarization, which opens more gated sodium channel, okay, or which opens more sodium ion gated ion channel, and this will allow more sodium ion to diffuse into the exoplasm, okay, either to diffuse into the exoplasm or to enter the exoplasm, or more influx of sodium ion into the exoplasm. Now, the stronger the stimulus, the more sodium ion gated ion channel will open. The strength of the stimulus or potential difference, okay, must be more than a threshold level. Okay, now for human, mostly the threshold level value is negative 40 millivolt. So here, okay, so that there is enough sodium ion gated ion channel open to produce an action potential. Okay, now to simplify this, okay, the threshold value, okay, is negative 40 millivolt for a chain reaction to start, where more sodium ion gated ion channel will open to produce an action potential. Okay, to summarize, in depolarization, only some sodium ion gated ion channels are open. Okay, but most sodium ion channel are open. But for the um, potassium ion gated ion channels and the potassium channel both remain closed here. Okay, in depolarization. Now let's take a look at the third stage. The third stage is known as the rising phase of the action potential. So what happens here? Okay, when the threshold from the second stage is crossed, okay, and the value of the potential difference is about positive 30 millivolts, an action potential will be generated. and sodium ion cathode ion channels will be closed. The potassium ion cathode ion channel remains closed. Okay, now to summarize, in the rising phase of the action potential, okay, both potassium ion cathode ion channels and potassium ion channels are closed. Okay, similar with the second stage. But most sodium ion cathode ion channels open to allow more influx of sodium ions. But these sodium ion cathode channels will close when action potential is already generated, which is here at the peak of this graph. Okay, so I need to say when action potential is already generated, okay, most sodium ion cathode ion channel will close. But, or sorry, not but, okay, while the sodium ion channel, okay, again similar with the sodium ion cathode ion channel, okay, most sodium ion channel will open and close when action potential is already generated. So in this region here, okay, sorry, this region here, most of the sodium ion cathode ion channel and sodium ion channel will open, allow more influx of sodium ions into the exoplasm. Okay, now when action potential is generated here, what happens? All channels are closed. Okay, sodium ion cathode channel, sodium ion channel, potassium ion cathode ion channel, and potassium ion channels are closed. Now, the fourth stage is the falling phase of the action potential. Okay, so what happens here is repolarization occurs immediately after the action potential. This will cause the nerve cell to be polarized, or this will cause the axon or plasma membrane of the axon to be polarized. Okay, now how does repolarization cause the plasma membrane of the axon to be polarized? Okay, first, sodium ion cathode ion channels are closed, right? It started from here, right? Okay, it's closed, so meaning to say this will prevent the inflow of the sodium ions, or it will halt the inflow of the sodium ion from the extracellular fluid into the exoplasm. Then most potassium ion cathode ion channels will open, and there will be a rapid outflow of potassium ion leaves the exoplasm into the extracellular fluid. So meaning to say the exoplasm becomes negative again. Then potassium ion channel are closed or inactivated, but most potassium ion channel open. So basically what happened in stage 4 is the inverted on what happened on in stage 3. Now, the fifth stage is known as undershoot. So what happens in undershoot? The axon membrane's permeability to potassium ion is higher than at resting phase. Okay, so this was the resting phase, and this is the undershoot. So what happens at the undershoot? The axon plasma membrane permeability to potassium ion is higher. So what happens is that more potassium ion will flow out from the exoplasm, causing a momentary hyperpolarization. Okay, the potential difference, okay, across the plasma membrane of the axon is more negative than the resting potential. Okay, that's why you have this value here. Okay, the membrane potential is more negative than the resting potential. Resting potential should be here. The sodium-potassium cation channel remain closed and the membrane potential returns to the resting potential. Summary: most potassium ion cation channels are open, okay, in the undershoot, but close when the membrane potential returns to the resting potential. The same goes for the sodium ion channel that is closed but open when membrane potential returns to the resting potential. Sodium ion cation channels remains closed and some of the potassium ion channel opens. Okay, so that's about it on the formation of the action potential. Okay, bye.